Okay, thank you very much. I would like to thank the organizers for organizing this and for giving me the opportunity to talk a little bit about our work together with the mathematician that you're going to hear from Magali later on. And I think this is a wonderful idea to pair up the experimental talks with the mathematics talks, just to showcase what can be achieved when experimental scientists like myself work together with mathematicians. Okay, so the topic of today is the experimental analysis of the division of amyloid fibrils. So I'm going to basically talk about amyloid fibril fragmentation. I'm going to talk a little bit about how we got into this area, a little bit about why this is from biological and biomedical point of view, why this is an important area. And I'm going to show a little bit about our latest work before I'm passing on to Magali, who is going to talk about the theory development behind the fragmentation work that we're doing together. So before I start, I would like to thank a lot of people that has been working with this project or collaborating in general with my lab. I'm going to highlight the mathematics friends that I'm working with. So that's Marie, Magali, and Miguel. So thank you very much for really working together on this topic for a number of years. I really enjoyed working with you guys. So we already heard a little bit about prions yesterday from Human's talk, and I'm gonna just say a few little bit about amyloid. So amyloid is basically filamentous protein assembly. So they all share cross-beta structure, but what's important is that they are basically filaments with a beta structure, but they all look different. And the different type of these amyloid filaments, they are associated with possibly different disease, but some of them are have biological function as well. So on this slide, I'm just showing you a few how the structures, a few of these amyloid structures, how they could look like if you imagine the filaments are going into your screen. So this is basically the cross section we're looking at. So as you can see, they're all filaments, but they all look, they can look very different. How do they form? So these structures are being formed when monomeric proteins, they come together and assemble into filaments. And this assembly is actually very slow, which is great in terms of disease association, because we don't obviously get these diseases until we are old. So these are all age-related diseases if the amyloid are associated with the disease. In any case, so when the monomers, they come together and form amyloid fibrils, they have to cross the so-called energy barrier, and then therefore the process is takes time. And you can also form a lot of different types of fibrils, even from the same monomer. Like what I'm showing here, basically the energy landscape, so-called energy landscape, is quite rugged in some ways. And if you look at the kinetic process of amyloid formation, we can typically follow it, for example, with fluorescent dye. Then we notice that these reactions, they are so-called nucleated polymerization reactions. What that means is if you start off with the monomeric protein, you end up with a long lag phase where very little is happening. And then all of a sudden, the polymerization reaction will kick off, and then you get this type of sigmoidal shaped curves when you characterize the formation of amyloid fibrils. And a characteristics of the nucleated polymerization reaction is that you can see the reaction with preformed aggregates. And if you add seed, so that's preformed aggregates, into a reaction with new monomers, then you can get rid of the lag phase, because the monomers can then build straight onto the seeds, which get rid of this barrier, so to say. So if you can see the underneath nucleated de novo reaction, we have the seeded reaction, which then goes much faster because there is no lag phase anymore. We are, of course, interested in amyloid for different reasons. So traditionally, people were looking into the formation of amyloid fibrils because they are associated with a lot of neurodegenerative disease, age-related disease, such as Alzheimer's disease, Parkinson's disease, etc., etc. But of course, disease association is not the only reason, because a lot of amyloid are associated with biological function, and then more and more are known about these type of functional amyloid. But of course, disease association is still very important. And as we heard yesterday, so prion disease is also very prominent feature, because prions, in a way, they are basically transmissible amyloid. And the transmission, as I'm going to talk a little bit about that later, is happening when you have tiny fragments of these fibrils. And that's basically one of the reasons why fibril fragmentation could be important also for prion disease. Of course, in terms of disease association, so we are all living in a very much aging society, and Alzheimer's disease, for example, here is important societal issue with the aging society. So I'm just looked into this on, for example, the Alzheimer's Society's, so in the UK's webpage, they have some infographics about the cost in terms of monetary cost and human cost of Alzheimer's disease. So as you can see here, it's very much an important and issue, and it's just going to get more and more important as the society age. In terms of the average life expectancy also obviously is growing. So it's very important to address this issue, not just fundamental biological point of view, but also from biomedical point of view. Now I'm going to talk a little bit about how we can study amyloid formation using experimental approaches. So one of the things you can do is monitor the formation of amyloid, and you can do that by watching how amyloid fibrils form using a fluorescent dye that binds only to the amyloid form. And then what you can see is you get these type of sigmoidal shaped curves, like what I talked about. So these are nucleated assembly reactions. And this reaction is highly monomer concentration dependent. So the higher, the larger the amount of monomers that you get in the solution, the quicker the amyloid fibrils will assemble. So another way to directly look at the filaments that's being formed is using microscopy approaches. So we can do, for example, electron microscopy or atomic force microscopy. So these microscopy methods allows visualization of the filaments that's being formed. And these filaments are in the order of 10 nanometers wide. So a nanometer is a billionth of a meter, so they are really, really tiny and far too small in order for us to see them using conventional optical microscopy. So we have to use these nanoscopic approaches. So with the AFM, we can, for example, see that the filaments, they are very different. Even from the same monomers, you can form different type of filaments. And as later on, I'm going to show you that you can also study this type of reaction as function of time by, in our case, taking snapshots of the filaments as they are being fragmented. So over the years, we are now knowing more and more about the mechanism in the processes involved in the formation of amyloid by studying the filaments using microscopy approaches, and as well as studying the kinetics of the formation. So now, if we boil down the key mechanisms, the key processes in the mechanism, then we have 4 different key processes. The first one is the primary nucleation. So this is when the monomers initially get transformed into the amyloid state through the nucleation process. And then we have the elongation. So this is the growth process where monomers that keep growing onto the fibril ends. And then we have two processes that are called secondary processes. One of them is secondary nucleation. So this is a heterogeneous nucleation, i.e. a nucleation process catalyzed by surfaces. So in this case, it can be catalyzed by pre-existing surfaces on the pre-existing aggregates, filaments. And the topic that I'm going to be more focused on today is the division process, or also called breakage or fibril fragmentation. So this is the process highlighted in yellow on this slide. So this is when larger filaments break and then form more smaller fragments, and these fragments obviously can continue to grow. So it's almost like bacterial cell division in some ways. So now I'm going to focus a little bit about why the division of amyloid fibrils, the fragmentation of these filaments, are important to understand. If you look at the amyloid life cycle, as I would like to call that I just showed you on slide, then the growth process by elongation is important, and this is also explaining why you can seed, so bypass the nucleation phase of the assembly by adding preformed fibrils. So in this case, if you have exactly the same amount of protein in mass, but your filaments are of different size, then of course, if you have smaller filaments, then you can have more of them with the same amount. And because each fragment can seed on their own, so therefore, the smaller the filaments, the more of them you have, then the faster the seeding process will proceed. So this is exactly what we see in now very old experiments. People have done that for a long time, and so did we. So this is, so you can see from my work when I was still a postdoc back in 2009. So if you add exactly the same amount of protein, but seeds that's much more fragmented, the seeded reaction will proceed much faster to completion. So another thing that back then we looked at is that so these amyloid fibrils that we can form, they do interact and perturb cellular processes. So if we can measure how the viability of cells change when you add fibrils of different from different reactions, then we also notice that in the same way, if you fragmented the fibrils to with exactly the same amount of protein, but the fibrils are much smaller but larger in number, then they are much better. So the same solution then will be much better at reducing the cell viability. So in this case, if you see the fragmented fibrils, in this case, lower the viability much more than the same amount of proteins as fibrils. So that's very important comparison, because that suggests that fragmentation, if you fragment fibrils a lot, then they might give you, they might have some kind of a cytotoxic potential, and the potential then depends on how much they're being fragmented, because it's an issue of number of filaments as well as what type of filaments, how many of them you've got, basically. So one of the possible mechanisms of disruption by amyloid fibrils is through disrupting membranes. And back then we also looked at, with these type of samples, exactly the same amount of protein, but with fibrils either fragmented or not fragmented. Then you see that if you have fragmented fibrils, so that means they are much smaller but much larger in number, then they are also more more efficient in disrupting membranes. In this type of membrane assay, the so-called dye release assay, we can see that the dye is being inside these vesicles are being much more released when incubated with the fragmented fibrils. So together with with cryo-EM lab. So in this case, Helen Sable's lab in London, we looked at how these fibrils are actually interacting with vesicles. And we did see that the interaction is happening mostly at the fibril ends. So in this case, if you have fragmented filaments, then you have more of these surfaces that tend to be more reactive. So that could be another way the division process might be important to look further. So in this case, so I'm showing you the EM picture when the vesicles are being put together with the long filaments, compared to if you look at when the vesicles is being incubated with very short fibrils with a lot of ends, then you see that the fibril ends are the ones that disrupts how the membrane look like. So this is all back then was very, very exciting. That suggests that there is a direct link between the cytotoxic potential of amyloid fibrils compared to, if you compare fragmented, not fragmented, then the cytotoxic potential is being changed, even if you have exactly the same type of filaments of the same amount. Like what I said in the beginning, that so prions, as far as we know, they are also basically the filaments that we look at in the prions. So they are amyloid filaments. The prions, they are just amyloid filaments that can be transmitted, like what we heard in the talk yesterday, Huma's talk. So prions can also exist in yeast. So we use that as a model system to study how prion particles can be basically taken into cells, basically in order to look at the infectious potential and how that is being affected by fibril fragmentation. Because the idea is if you fragment the filaments, then you have larger number of them, which already is going to obviously enhance the infective potential because you have more of them, but also you get smaller fragments, and smaller fragments will be more likely to enter the cells in this case. So we use, like I said, yeast prion as a model system to study this process. So the yeast prion we're using is the amyloid form of a protein called sub-35, and in yeast, in the amyloid form, so sub-35 is forming aggregates, and we have a model system. Whereas if the protein is in its amyloid form, basically it will give you a prion phenotype, in this case psi plus, and attached with it is a colorimetric change in the cells. So in this case, to cut a long story short, we see white cells, white colonies instead of red cells. So we can distinguish when the yeast cells are either in the prion negative state or the prion positive state. So we did this using in vitro generated sub-35 amyloid fibrils. So we can fragment these fibrils to different extents, and we can characterize their size distribution, which also then gives you an indication of how many particles you've got, because you know how much protein you put in. And then we incubated these samples with the E cells just to see how many E cells, when incubated with these filaments, then going from a prion negative state to a prion plus state, by looking at the color, like what I just said. So we done it with a lot of different samples. And we then characterize the colors of the colonies that's being formed. And we can see a correlation. So as expected, the more particles you've got, the more likely is you then get a prion plus conversion. But if you just look at the number, it's not enough to explain, because if you have large number of large particles, it doesn't actually do very much. So the conclusion from this study, which it was done already a couple of years ago, is that that two identical particles is not equally as active in terms of how well they are in basically infecting the yeast cells, in this case, infecting. So the concentration of particles is one parameter, but the size is also important, because you can only convert the cells to pre and plus if the particle is less than a certain size. So we looked at basically a model describing this process using a threshold model. So and we think a threshold around 200 nanometers is basically the estimate of when it's now starting to be able to get into the cells. So all of these is really, these are all the experimental work we've been doing for over 10 years, and we've seen that all of these processes will be affected by fibril fragmentation, because amyloid structures, they can be either large superstructures, which means they're actually not that very active, they could be inert, they don't get anywhere, but they can also be very tiny particles, so they are the nanoparticles that can be very active biologically. So we really need to understand fibril fragmentation a bit more. And so basically then the question is, how do these particles divide? And the division process is essentially a very big part of the growth process that's happening in large quantities in the middle of the growth curve, in these sigmoidal curves. So this, we also did some simulations to show that if you look at the long time scales, then this is actually the process that's dominating growth, especially if you have limited amount of monomers. So this is in vitro for experiments, it's super important. So we're starting to design experiments to look at fragmentation on its own. So what we basically devised is we want to be able to just study the fragmentation in the absence of any growth. You can do that in a test tube by basically using already formed fibrils, and then you can perturb the fibrils. So normally fragmentation takes a very long time, but we can accelerate the process by, for example, putting on some mechanical perturbation. And then we can take snapshots of the pictures using the atomic force microscopy methods that I just told you about. Then we can see that we have a time course, essentially, looking at fibrils that's going from fewer but longer to much more in number but smaller. We can characterize the size distribution of these filaments, and then we can basically see how the size distribution, so the population of sizes, how that change as function of time. So this is experimental work before I started to work with Marie Magali and Miguel. So from these reactions of one protein, we saw that the fragmentation seemed to be very much size dependent, which is something that people haven't considered back then. And I think it's now widely accepted that this is the case for amyloid fibrils. And then we also did time course, and when we compare, when you have fibril fragmentation as with experiments where you have both fibril fragmentation and growth, we saw that growth fragmentation for fibrils, they have complicated processes because you get biphasic behavior, since the rates of growth and fragmentation, they are in different regimes. And this is far too complicated. So we basically, it's a confirmation that it's really good experiments that we first devised, that we only look at fragmentation in order to understand the fragmentation process a bit more. So now I'll come to our recent work, which we're going to hear much more from Magali. Basically, we looked at how we're going to compare fibril fragmentation properties if we have amyloid fibrils of different kind. So in this case, we've done this type of fragmentation only experiments with different systems. One of them is alpha-synuclein, which is disease-associated amyloid. So the same type of time course. And then we characterize the change in the size distribution, so length and height. We saw that the length distribution changed a lot over time as expected. So the questions then are, basically, now we know fibril fragmentation rates are length dependent. Can we do experiments that are comparable? So independently of which type of fibrils and which starting point you start with, which is very hard to control in experiments. Can we do something to be able to compare the fragmentation properties of different fibrils? And can we extract meaningful intrinsic properties of amyloid assemblies? So the answer is yes. To cut a long story short, so the conclusion we got from this is alpha-synuclein fibrils. Now this is the disease-associated system that we look at. It tends to be less stable toward breakage. So it breaks much more with the same, if you consider the width of the filaments especially. And they are more likely to shed smaller particles from its fragmentation process. So if you look at the distribution, you tend to have more smaller particles. So with that, I'm going to pass on to Magali, who's going to describe. So basically, I just told you from this analysis that we've done together, what kind of conclusions we got. So now I'm passing on to Magali. She's going to tell you all the details about how we developed the theory, how Marie, Magali, and Miguel developed the theory, I should say, and how then we together applied onto data to actually extract that information that turned out to be really biologically relevant. Okay, thank you very much.
Hello. Thank you, Wei-Feng.
Thank you.
I'm trying to share my screen.
Yes, we can see there is a line in the middle, a green line.
Yes, I don't know why. So, okay. Back. Still a green line?
Yes, it's still there.
Okay.
I mean, we can see the slide. I don't know why it's appearing there.
I don't know. I can try one more time, but I don't know.
But by the way, we hear you a bit. The sound is a bit low. I don't know if you can raise a bit.
I cannot do. Okay, what I can do is that. Is it too much?
Too much. Yes, you can put a bit less because there's a lot of sound. Okay, try.
Like that.
I think it's better. Yes. If not, we can raise the volume. Okay, I think that's good.
Okay, I will try to speak louder.
No, don't worry. I think we can also put the volume up. That's fine.
Sorry for the green line. Actually, I see it as well. My screen seems to be divided into two parts.
Magali, maybe if you activate full screen first and then share the full screen window.
Okay. Yes, but the problem is that I cannot do it. I need to not to be full screen to have access to the button share screen. Okay. Bon. Sorry for the green line, and I hope you will hear me.
Yes, don't worry. We can see well the slides.
Okay.
Thank you.
Yes. Thank you for the invitation. Thank you, Wei-Feng, for your talk also. And yes, so what I will try to do is to detail how we obtained the results that Wei-Feng presented, the last result that Wei-Feng presented about the comparison between different amyloid fibrils. So for instance, alpha-synuclein and beta-2M, as he presented. And the model we used to describe the experiments of Wei-Feng, in agreement with him, is a so-called fragmentation equation. We use this fragmentation equation in a continuous setting. We could have used it in a discrete setting, but it was better for us to do math on the continuous setting. And since the experiment didn't go too far in time, we did not have too small particles, so we agree that the continuous setting was precise enough to describe the experiment. So we also agree on this setting. And so the question is, we will have equation, and we want to characterize the fragmentation rate, the fragmentation kernel, and each particle has its own fragmentation rate and its own fragmentation kernel. And we want to determine it for each particle. So again, this is the same work, so it's a common work with Marie Dominique, Miguel Escobedo, and Wei-Feng. As an introduction, I will talk about the fragmentation equation. I will present the equation and present its basic properties. In particular, its asymptotic behavior, because based on this asymptotic behavior, we obtain some results. Then I will formulate the inverse problem and detail what kind of mathematical result we obtain for this inverse problem. And at the very end, I will explain you how we did recover the parameters in practice. And part 2 and 3 are a little bit different. Okay, so the fragmentation equation, so it's equation whose unknown is the density of amyloid fibrils here, of size of length x at time t. So the number of particles is decreasing because particles are breaking into two. So this is this term, and they are breaking into two with some fragmentation rate b of x. The classical assumption, which is made by both biologists and mathematicians, is that b of x is actually monomial in x. So as Wei-Feng said, it depends on x only. And so to recover b, we need to recover two parameters, namely alpha and gamma, and they both are positive numbers. And we have creation of particles of length x. It happens when a particle of length y breaks into two particles, one of length x and one of length y minus x. So this is just an equation who says that particles are undergoing fragmentation and that everything is preserved. By everything, I mean the total number of monomers is preserved. Some assumption about the fragmentation kernel, which is also classical. It's assumed to be a self-similar fragmentation kernel. Namely, it means that if a particle of size y is breaking at the probability of a particle of size y breaking at the locus x only depends on the ratio x over y. So for instance, if small particles are only breaking in the very middle, it means here that kappa is a delta function in 1/2, then large particle also will only break in the middle. And the three equations I wrote at the very end of the slide, I will focus on the second one. The integral of kappa is equal to 2. This is an important assumption. It means that particles can only break in the experiment in 2 smaller particles, not in 3, not in 4, or not in 1. Okay. Brief history. This equation was actually introduced by Kolmogorov. He was describing stones which were undergoing some grinding, and he realized that the size distribution of particles tends to distribute to some distribution. In his work, the fragmentation rate B was not depending on X, it was a constant. And at the very end of its paper, he was saying, well, it would be interesting to study this equation with very generic fragmentation rate and very generic fragmentation kernels. From the 1940s, we have a lot of literature in physics about polymers fragmentation, where people are using this equation more in the discrete settings. And more recently, in mathematical biology, this equation is used to describe so amyloid fibril, but also cell division and bacteria division. Okay, very basic properties. So this fragmentation equation has a global solution. And we have the conservation properties, so the mass conservation, saying that the total number of monomers is preserved, and the total numbers of particles is increasing. And we have that the total number of particles, its derivative with respect to time, grows as the integral of bf. Okay, to talk about the large time behavior of this fragmentation equation, I will talk first about the large time behavior of the growth fragmentation equation. It's the same equation, but particles can fragment, but can also grow. Actually, in the experiments of Wei-Feng, we could have incorporated the growth term, but we agreed that growth was negligible in the experiments. But if we have some growth at rate x, then it's proved that this equation, so its solution f, converges as t goes to infinity to some profile, capital N, and this result is based on spectral theory. Okay, so now what about the asymptotic behavior on the fragmentation equation? As Wei-Feng mentioned it, particles are becoming smaller and smaller, and you see it on the pictures of Wei-Feng. Remember the brown picture with the white particles. At early times, we have a small amount of particles, but they are very long, and at large time we have lots of particles and they are very, very small. So since particles are only undergoing fragmentation, this is expected. And actually what is expected and what is true is that f of tx, the density of particle, the size density of particle, is going to some delta function at x equals 0. At the very end, at time c equal infinity, we only have particles of size 0. But the thing is, it goes to a delta function in 0 with some given profile. So the theorem is as follows. For large time, the profile f of the size distribution tends to distribute according to some, we will call it some asymptotic profile g. Or in other words, if we perform a change of variable, f of tx tends to g. The proof is based on the change of coordinates. If you make the clever change of coordinates, the fragmentation equation turns into a growth fragmentation equation, and then you apply the result of the growth fragmentation equation for the growth fragmentation equation. And you have the equation, and this equation we will study it later. So this equation on the asymptotic profile G, this is the stationary equation that I wrote here. Okay, I will illustrate this theorem. So here I picked some parameters, gamma equal 1.3, alpha equal 1, and kappa is a two-peak Gaussian. And we have an initial condition, which is a spread Gaussian. So let us look at the picture on the left. So you see for different times, from 0 to 18, the distribution f, and it's maybe not easy to see, but this profile tends to converge to a delta function in 0. And what happens is that after some time, it will always have the same shape. And if you look at the picture on the right, you have the same numerical experiment, but in rescaled coordinates. Now you see it very well that starting from time, say 13, it seems that the profile, the rescaled profile, has converged to something. It is one peak distribution. Okay, so now what is the inverse problem? Wei-Feng provided us some data, and the data looks like the picture on the right. So we have, for some time, the size distribution profile of one given particle. And the parameters gamma.
Wait, Magali, we cannot hear you. I don't know what happened. Okay, now we can hear you. I don't know what happened. I don't know if it was me.
It may be my Wi-Fi, not Wi-Fi, actually, my connection. Yes, so So this is the kind of data we have, and from this data, we want to recover alpha, gamma, and kappa. So you see on the data of Wei-Feng, they really look like these numerical experiments in the sense that the profile tends to reach a stationary profile at the end. Okay. So now I will focus on the uniqueness and reconstruction formula. For that, I need to introduce the tool we used. This is the Mellin transform. The Mellin transform of some measure mu is defined as, so the Mellin transform of mu at S, it's the moment of mu of order S minus 1. I write it very heuristically here. I do not put spaces and everything. Okay, the Mellin transforms looks like a Fourier transform, except it's not exactly the Fourier transform. But we have similar properties. So I will start with property 2. If the Mellin transform exists for some real number A and B, then it will, for some measure mu, then it will exist on the compact set AB included in R. And it will also exist on the vertical strip, which is delimited by A and B. So typically, Mellin transforms of measures or of functions are defined on a vertical strip on the of the complex plane. We have the equivalent of the Riemann-Lebesgue theorem. So Riemann-Lebesgue theorem for Fourier transforms say that if the function is L1, then its Fourier transform goes to 0 at plus and minus infinity. For the Mellin transform, if the measure has a density F, which is L1, then its Mellin transform goes to 0 as its imaginary part, V, goes to plus or minus infinity. We have an inversion formula of the Mellin transform. And if you know the Mellin transform capital F of density small f, then to recover small f, you need to integrate capital F on a vertical line of the complex plane. And here on the formula, the vertical line has some real part Q star. So just to mention it, the right-hand side does depend on Q star, whereas the left-hand side does not. Actually, this formula, under some assumption, does not depend on Q star. And this is because of the Cauchy theorem. The integral on some vertical line does not depend on the vertical line, because the integral of on a closed path on the complex plane is 0 if the function goes to 0 at plus and minus infinity. So that's the idea. Okay, so the Mellin transform, we used it to turn our linear integral differential equation, whose solution is a stationary profile G, into a functional equation on the Mellin transform of G. So I define capital G as the Mellin transform of G and capital K as the Mellin transform of the kernel kappa. So now if we look at the equation, so D1 is just the domain of kappa and of K and G. If we look at this equation, we can see it twofold, in two different ways. First, if I give you the parameters alpha, gamma, and kappa, then you can compute K, the Mellin transform of kappa, and you can solve this functional equation, find capital G, use the inverse Mellin transform, and find the stationary profile small g. Or if you know alpha and gamma and the stationary profile small g, you can compute capital G, its Mellin transform, and then get K from the equation and recover the fragmentation kernel. So this equation will be crucial in our inverse problem. Okay, the theorem we got is, let's take some profile G, which satisfies some assumptions. Then we can find at most one triplet, alpha, gamma, and kappa, such that G is the solution of this stationary equation that I wrote before. Of course, we don't have existence, which means that if I take any random function in L1, it may not be the stationary profile of some fragmentation equation. But we have uniqueness of the parameters. I will talk a little bit about the uniqueness for gamma and alpha here, and kappa also. So we have two proofs for gamma. They are equivalent, except that one is under more restrictive assumptions, and it doesn't rely on the Mellin transform, and the second one does rely on the Mellin transform. Let's look at proof number one. Some authors have proved that the asymptotic behavior of G, by asymptotic, no, I mean the behavior when x goes to infinity of G, was driven by gamma, in the sense that the log of 1 of G was behaving like a constant multiplied by x power gamma. So now we know from that we deduce that if we know G, the stationary profile, then looking at its behavior for large x will give you gamma. So you know gamma. Then the functional equation that I wrote here, if you plug in it s equal 1, and then that you use, that k of 1 equal 2, I recall that k of 1 equal 2 comes from the fact that the integral of kappa is equal to 2, which comes from the fact that we only allow particles to break into 2. Then you find alpha as a function of gamma and capital G that can be computed from G. And the uniqueness for kappa now, okay, we know capital G. We know that G is not 0 everywhere. So capital G, for S, a real number, a real positive number, is strictly positive. So we can define K of S with this formula in red. I just work on the equation and reverse the terms here. So that means that this formula makes some sense. So kappa K of S, which is equal to the Mellin transform of kappa, can be anteriorly determined. And then we can have two arguments. Either we can interpret K of S as the Laplace transform of another function and use the uniqueness property of Laplace transform for measures. Or what we can say is that K is holomorphic. We determine K on a compact set of R, and using the isolated zeros of complex holomorphic function, it means that K is determined on the on the complex plane, actually here, and so in particular on any vertical line. So we can use the inversion formula and get kappa. Okay. Now we wanted to have a reconstruction formula for kappa, because this cannot be used. You know, you need to have to use a theorem that extend k from the real axis to the whole complex plane. So what we wanted to do is to define k as this, but for s, not real number, but s on a vertical line. But the problem is that capital G, we do not know whether it can cancel or not on the complex plane. And in particular, we do not know if we can find at least one vertical line on which capital G never cancels. And actually, our first theoretical paper, most of the paper, what about this very delicate point, prove that we can find a line on which capital G does not cancel, and then define K and prove that everything works. So this is the theorem that we got. Actually, we proved a little bit more. We proved that capital G does not cancel on a vertical strip. So we proved that we were able to define K on some vertical line included in this vertical strip. And then that the kappa we found with the inversion formula does actually correspond to the real kappa we were looking for. This I don't talk about. So conclusion, we are happy because we are sure that there is only one solution to this inverse problem. We got a reconstruction formula for kappa, but we do not have a reconstruction formula for gamma yet. And I wanted also to say that the way we extracted gamma from G, from the stationary profile, depends on how G behaves for large sizes. And this means that we need to access moments of high orders of G. And this is very difficult in general. So now, how we do recover the parameters in practice. For the theoretical parts, we only use the asymptotic profile. But we need to recall that Wei-Feng provided us more data. It provided us the profile for different time. And actually, we can determine gamma from the history of the experiment. So how do we do that? So, okay, I said that the fragmentation equation was preserving mass. Actually, this is not completely true. I mean, this is true, but what we access with the Wei-Feng experiment is not the total suspension. Wei-Feng is taking some aliquots out of the suspension from time to time. And each time he may take some sample with different masses. So what we access is not actually F, but it's U, which is the density, the probability density of the size distribution. So now if we define M1, which is the mass of U, and assuming that the distribution F is close to the equilibrium, it means that F is equal to T power 2 divided by gamma G of T 1 over gamma X. So U, just using the formula above, is equal to T 1 over gamma G of T 1 over gamma X. And if we multiply by X, integrate on R plus, and pass to the logarithm, then we obtain that the logarithm of M1 of T is expected to decrease linearly with respect to log of T, and the slope is 1 over gamma. So this is great because then we just have to plot log of M1 of T at the function of log of T. If we do not obtain a line, then it means that either our model is not good, either the experiment actually was only done for early times. And if we find a vertical line, we can say that this slope is 1 over gamma. So let me illustrate it on simulated data. So this is gamma equals 0, 5, 1, 2, 3, 4. This is a plot of the log of M1 of T, and you see that for large time, we have a vertical line, and the slope is exactly minus 1 over gamma. So now how do we do it for real data? Okay, yes, here. Real data actually look like that. Why do we have many lines? Because here we do not plot only M1 of ST, but M of ST for S. If you want to look at M1 of T, I think it's the curve, the line below. So for A in 6, it seems to be a straight line, but for beta axis, it's not a straight line. So the idea is what protocol will be used to recover gamma. And the protocol we decide to use is as follows. So I illustrate it on simulated data. So on the left, I have simulated data, and on the right, I plotted as a function of time M1 of T in log-log coordinate. And you see the data points I have are in red here on the pictures. So if I interpolate linearly, I have the curve in blue. It looks like a plateau and a linear function. So what we said is that, well, we have some reason to think that it's often behaves like that. So we assume that the profile M1 of T as a function of time looks like a plateau and a vertical line. And we use least square minimizers to fit the data and to get the slope. So that's how we got gamma. Then we did some numerical investigation on how robust this method is. And of course, we found that the more late time data point we got, the more precise we are on gamma. And we also explored the sampling noise. So let us assume that we have access not to the entire density, but to a sample of size, let's say 200. So in this picture, the real data is 1. It's in red on the curve. The estimated gamma is in green. It's the estimated gamma using this method when we access the complete distribution. Now, if I make an experiment, a numerical experiment, when I take a sample of 100 particles, and from these 100 particles, I reconstruct the density. It will be not a precise reconstruction, but it will be some reconstruction. And then from this reconstruction, we try to get the estimated gamma. You see that you can obtain all the points in blue. And if you average them, you obtain the curve in black. So here, what we can say from this plot is that if we make an error of 10% of gamma with the complete distribution, with 200 particles, we have an error of maximum 20%. So we explore this more precisely. Then we also explored how gamma was robust to, how the estimated value of alpha was robust to gamma. If gamma is perfectly found, then gamma alpha is also. But if gamma is wrong, alpha is wrong also. But alpha x over gamma may be good for the average size x. And now about the kernel, I will show two plots. So the plot on the left, we have a lot of different kernels, and you see that the rescale distribution profile that correspond to these kernels are very similar. So they can be classified into two. Kernels would create particles of very small size, so whose kappa of 0 is something strictly positive, and kernel whose we are creating very we are not creating small-sized particles. And you see on the picture on the right, it's even worse. If you compare a kernel creating small-sized particle and no middle-sized particle, and a kernel creating small-sized particle and a lot of middle-sized particles, then the rescale profile are the same. So the conclusion is here, okay, our reconstruction formula cannot be used in practice because we cannot, distinguish kappa on the asymptotic profile. We quantified it, and we make the assumption that, okay, let's assume that we start with the same distribution. Let's assume that we have gamma equal alpha, but two different kernels. Let's say, for instance, on the figure on the left, the kernel in red and the kernel in blue. Then, can we say that the distribution at time t are the same or not? And you see that if the initial condition in red or in blue are anything but a peaked Gaussian, which is in black, it's impossible to distinguish from some distribution the different kernel. The only distribution that can be used is a very peaked Gaussian in black. So if you start with a very peaked Gaussian in black, and if you have access to very early time points experiment, then you can hope to distinguish one kernel from the other one. So I will stop here and conclude that so to determine gamma, we need several data points for large time. To determine alpha, we need at least 1 point for large time. And for kappa, this is still an ongoing work, but we find that we need early data points for a very specific initial condition. And this initial condition corresponds to a monodisperse suspension, meaning that all particles have same size, and it cannot be obtained in practice. So we need to work a little bit more to get kappa. Thank you.
Okay, thank you very much. Okay, so thank you for your talk. We are going to take questions. As always, please unmute yourself and go ahead or write on the chat.
I have a questions for Wei-Feng and Magali. A little bit more focus on Wei-Feng's data. I would like to. I have in fact two questions. Wei-Feng, do you know if you see any correlation between fragmentation rates that you observed and the young moduli of fibers that I think that we can find it in the literatures for alpha-synuclein and tau and others fibers that you tried?
Yes, it's a really interesting question. So I have no answer to in terms of experimental evidence. So we did try to look into this, but we can't draw any firm conclusions regarding with the limited data set we've got. Then it does look like. So as you know, Young's modulus is correlated with the persistent length of the fibrils in some ways, and you can actually look at the persistent length of the filaments. But of course, the best way is to directly measure Young's modulus in some ways, which is not very easy. Yeah. It does look like beta lag, which is much stiffer. It has very long persistent length, and it's also the one that has the highest length dependence, so the highest gamma. So we didn't put any of this sort of thing in the paper because we are not sure yet how that will pan out, but it certainly is a very interesting aspect, because you can imagine that if you can find a good correlation, then perhaps the stiffness of the filament tells you whether you're likely to break and how small fragments you might get in this case. So yeah, thank you very much for that question.
No, I have another question, if you allowed me. Concerning, in fact, the size of the fragments that you observed is more or less in relation also with the first questions. But do you think, or do you measure if the length of the fragments that you observed, even if they are different from kind of fibers, is a harmonic of the wavelength of the ultrasounds that you used?
No, I have not done the experiment. So I should clarify that we use two different types of mechanical perturbation. So in the data set in the paper that I showed, and which Magali also talked about, we didn't use the sonication method, we used the stirring method.
Okay.
But the stirring has also a frequency. It's it depends on how fast you stir. For the sonication, the only data that's sonication linked is for the yeast prion work, where we basically for the prion, we just wanted to generate different samples. We didn't measure the time dependence, and therefore the easiest way to generate at short period of time is use sonication. So we do have, I believe we have the frequency information, but we only done it with one frequency, so we don't really know that. But all of these ideas are really interesting. I think now that we find a way together, with Magali, Marie, and Miguel to extract those parameters. If we do enough of experiments and look how the parameters will change, then perhaps it will tell us a bit more about the type of fragments that's being formed, as well as how often those fragments are being formed.
Thank you. Thank you very much.
Thank you very much, Fuma, for the questions.
Thank you. Any more questions? Please go ahead or write on the chat. Okay, in the meantime, I have a question for Magali. I'm curious, do you have some kind of intuition to say, you use this Mellin transform that I'm not used to it? Why it's the right tool to, I mean, clearly at the end you see that is the right thing to look at, but what is the intuition to say, okay, here I should use this transform?
Okay, when you have a convolution product, you use a Fourier transform because Fourier turns convolution into a product. Here we do not have a convolution product, we have a multiplicative convolution product. We have f of y multiplied by k of x divided by y. And Mellin transform turns this multiplicative convolution product into a product. So this is the main reason why Mellin transform is for this kind of equation, and this is due to the structure of the equation.
Okay.
May I ask perhaps a very naive mathematical questions? Is Mellin transformations is kind of autocorrelations for process?
What do you mean by autocorrelation process?
In physics, we use autocorrelations to see if two phenomena covariance, kind of covariance is kind of covariance.
I don't know. I don't think so. Covariance of what? Because Mellin transform is a transform for one measure, only one.
Yeah,
so
but
it's just the moment, actually. You can see it at the moment of a function. So Mellin transform at s equal 2, it's the moment of order 1. Mellin transform at s equal 3, the moment of order 2. It's the function in the moment's coordinates.
Okay, I will go to look because I'm too naive for such things. Sorry.
No, maybe I misunderstood the question also. Because
it's me.
Okay, thank you. Any more questions? Okay, I have a question for Wei-Feng because I really don't know anything on amyloids. Maybe the first question is, so for this disease with Alzheimer and all this, I understand that amyloids exist there, and so when there is a disease, is because they stop working as they should work normally, or is that they appear the amyloid? I don't know.
Yeah. Okay. So, what I think your question is, how is amyloid associated with the various disease? This is actually a very controversial point, and nobody knows for sure. That's why I was very careful to say amyloid fibrils, they are associated with disease. Because one reason they could be associated could be that they are causing disease. The other one could be that they are the consequence of the disease. So it's not sure yet the cause, so the causality loop, if you will. So one idea is that when you have amyloid fibrils, they themselves are actually quite inert, but they then release these little fragments that can cause cellular damage in some ways. For example, by disrupting membranes. But this is not the only hypothesis, and all the hypotheses may not be mutually exclusive. There might be different ways of how this type of things can cause damage. And in Alzheimer's disease, it's also more than one amyloid that's being found. For example, we have the plaques. So the plaques are being, they are basically the aggregates from amyloid beta peptides. But we also can see the neurofibrillar tangles, which is formed by a protein called tau. So there's a lot of unknown of how these interplay with each other. And whether or not once you see the plaques, it's already too late. In fact, it's more intermediate that's perhaps more indicative of disease. So a lot of things are actually unknown. But we do know that amyloid is definitely associated with
disease.
Either, yes, exactly.
Okay.
Yeah.
Okay. Thank you very much for the clarification.
No. Thank you. I mean, that's why it's a very interesting research area, simply because there are so many unknown questions, and people tend also not to agree with each other because there are a lot of different ideas of how things are linked to each other. The causality loop is not known. So.
Okay. Yeah, it's fascinating. This. Okay. Thank you.